Yeah, so today it's about databases, data repositories, and standardization. And these are sort of uh, things that one are definitely necessary. It might not be the most exciting usually, but we'll need to go through some of it. So to start with protein sequence databases. So there are a few different databases that have been developed, and one of them is RefSeq. So it should be contain it should be a reference collection for different species. So it's has the protein sequences linked back to the genome, and so it provides a non-redundant database. And it's always a question of how you define non-redundant. And RefSeq for a very long time had uh, pretty much only one representative per gene, or maybe a few. Now, recently, a few months ago, they added on a lot more different splice variants. So the database grew quite a lot for, especially for human, and also for mouse. But for so this is it's being curated actively, and the idea is that it should reflect the current knowledge about sequences of the species that they support. So another one that comes out of EBI in the UK is Ensemble, which is very much connected to the genome projects, that they have a very consistent way to annotate genes, transcripts, and the proteins. And it's very nice because you can go back and download any version very easily, and it has the and from there easily get the mapping of the protein to the genome. So that's and they have they're working on more and more species to include here. Then there is UniProt, which is probably the best annotated. It's usually a slightly smaller set because they rely very heavily on human annotators. So they have like lots of people that spend their whole day looking at the literature and writing annotations about proteins. And like, I don't know if they have maybe 50 or 100 people that just do this every day. Then there are, of course, species-specific databases that if you work on a model organism, it's definitely best to go to and use the database that they can provide. And most model organisms have very good very well annotated databases. So uh, the the most common format for sequences is what's called the FASTA format, and it's a simple format in the sense that you have first one line that starts with the larger than sign and followed by an identifier, and then the identifier is anything that's comes until the first space. And then after that, you can write anything you want. And as you see, different databases provide slightly different formats. That here, for example, the species is within the square brackets. UniProt writes OS equal to the species, and so on. So pretty much after the space you can have anything, and here you have to have a unique identifier. And then you have one or more lines of amino acid sequence. And this is most search engines for to identify proteins take these FASTA files as input. And so then there has been suggested, since there is this freedom to write anything on that first line, people have tried to sort of make a more systematic way to write things so that one can actually use the information on that first line in an easy way. So this is so PSI has created this extended FASTA format where you can encode different information in a standardized way. So you can say that which residues are modified, and what's the species here, referring to taxonomy ID number instead. So you can, and this slowly more and more people are using this, but it's been very slow. And in general, it's still the just freer FASTA format that's used. Then, as we've discussed a little bit, one can also make sample-specific databases by generating next-generation sequencing data on the same sample. And that Kelly is going to talk more about on the 31st. So then, data repositories. So one big thing is that people generate a lot of data, and it would be very nice to reuse this data. There's been a lot of discussions that everyone should deposit their data and make it publicly available. There was quite a bit of resistance to that for a long time, because people felt that even though they published their first paper, they wanted to go back and reanalyze the data and get more information out of it, and they didn't want to make it public. But the thing is that it almost never happened that people went back and reanalyzed it, because you would generate more data instead and analyze that. So that didn't work out. So that was one problem. The other problem was that these data sets are quite big, so there was no good place to put it. And then what happened was that there was a repository created called Tranche that people started using, and it worked pretty well. And the journals started to say that one should deposit at publication. But then the funding for Tranche went away, and it still continued to work for a while, but then disappeared. So then the journals had to say, okay, there's nowhere to put it, so you don't have to make the data available. But now there are several repositories that are available, so now it's really not a problem anymore. So these different repositories have come together and created what's called a Proteome exchange. And the idea is that you can be a part of the Proteome exchange, and everyone within the exchange is supposed to share the data. So one of the biggest players here within Proteome exchange is Pride, which also comes from EBI. And they've set up so that people, when they want to publish, they deposit their data there, and they get an ID number that they can put in the publication, and then people can go back. And so what happened with Pride is that it was actually for a long time very complicated to submit something. They required a lot of metadata, which is great to have afterwards, but it really made it a big barrier for most labs to deposit their data. So then they came up with a more simplified format with just really the necessary information to be able to make sense of the data. And I think then now, and they also provided better tools to do the submission and description data. So now it's used a lot. The other one is Peptide Atlas, which comes out of the Institute for Systems Biology in Seattle. And they focused on making a repository where you could link the peptide sequences to mzML spectra that to see. And they also made special repositories for targeted analysis, so the SRM Atlas, and that's what you're going to hear about next week, more about how to do targeted analysis. Then we have Chorus, which is quite new player, and here it's Chorus provides a very nice interface in the sense that you can define your lab and set up what instruments you have, and then define different experiments and upload your spectra, and also do the analysis within the Chorus system. And if you do, of course, not if you do very special analysis, but if you do the some standard pipelines, and they're adding on more and more pipelines there. And the nice thing is that you can use this both as an analysis platform, and then when you're ready, you can make it public very easily. That part of the data that you want to make public. So this is something that's been growing more and more lately. So then again, there's another repository called Massive that comes from UCSD, and they also provide you with also a lot of tools and a lot of analysis. And there's so on all these, there's going to be more and more public data sets available. And then, so the one that I've been involved in is the global proteome machine, which, so the main person behind this is Ron Beavis at the University of Manitoba. And so he came up with this idea of that one should really collect, have a genomic focus, so that he built it on Ensemble. And then for every, you can dig down, look at the gene or a protein, and then dig down, see what evidence there is in the public domain. So right now, there are about 300,000 LCMS experiments that have been taken from the public space. So that's definitely the biggest collection of different of mass spec data. So the nice thing, so we can look at, so what one can do is one can use it, for example, to validate one's experiment. So here, let's say that we have so this is a linear representation of a protein, and the red is the peptides that we identified, and the dark red is the high confidence, and the pink is the lower confidence. And the green are parts of the protein that we don't expect to see because they're either very small peptides or very large or very hydrophobic. So what one can see here is that if we compare our experiment to everyone else's experiment, so these are the 20 top experiments where this protein was seen, we can see that it's a very similar pattern. So then we can, if we can of course be more confident that we have actually identified this protein if we see a similar pattern as other people do. We can also compare spectra, so we can start with our spectrum and compare it to spectra in GPMDB, and then we can see that there are roughly the same relative intensities is what we see in our measurement of a peptide compared to other people's measurements. So this is basically based on crowdsourcing. So any lab in the world performs an experiment, then they deposit it somewhere in a public repository, and then GPMDB imports it, runs some QC on it, and then either accepts or rejects it. And about 20% of data is rejected, and mainly because either the chromatography is not reproducible, that one can see that maybe the electrospray worked for a while and then in the middle it dropped, and then there was no data, or there's a big part of time where there's no data collected, or things that when the sample is carbamylated too much, which is usually from using old urea in the experiment. So things like that, those data are removed, but otherwise it goes in, and then it gets loaded into the public collection, and then people can look at the data. So here, the choice that GPMD made was that one should, there's only very little information that's required. It should really be simple to submit data. So main thing is the raw data files, or some other format of the data, and supplementary information is only if it's possible to get it. But usually it's good to know the species, but this can often be deduced from just testing different search, at least for the most common species. It's good if people provide a little bit more information, what how they did the experiments and what the reagents they used. Then, of course, it's helpful if there's information, if the samples were labeled with SciLac or iTrack and so on, and also what kind of enzyme was used. But again, if people use the most common, there are only a few, then this can be figured out automatically. Then we actually run into a different problem here, in a sense that now we've developed quite a good statistics for how to handle the case when we do one LCMS experiment, and we generate, let's say, 50,000 mzML spectra. But now in GPMDB we have 300,000 LCMS experiments. So it's a much bigger problem on, and if we would if we do the regular statistical filtering, we either we can filter too much, that because we have so much multiple testing that anything will be, or nothing will almost be significant, or we can run into the other problem that we let through too much false positive, and it looks like we have evidence for all the proteins in the database. So we've been trying to look at what's the how we can at least qualitatively characterize and divide protein into, if there is high evidence, I mean high confidence, medium confidence, or low confidence. And one way to do this is to look at the observations. So here is an example. So for P53 we have 212 peptides, and so this is, each row here is a peptide, and this just showed the distribution. So this is now how these peptides are distributed among among the different expectation value range. So this is between 10 to the minus 1 to 10 to the minus 2. This is 10 to the minus 2, 10 to the minus 3, and so on. So what we can see is that these are, of course, all very good hits. We can, what we observe is that usually we have quite big negative kurtosis, and the distributions are usually skewed, and we have also many observation overhold range. So this is another case then where we don't have very many observations where, for this protein. They're all at not sort of borderline expectation values, even though we have many peptides in the database. And then we have also another case where we see that we have a group of very good peptides, some sort of borderline, and then we have some bad ones. Even though we have several observations, we can then, by looking at the skewness and kurtosis here, we can say that even if we have many observations, it's not very likely that these are real. Then we can see that here we have a case where we observe a peptide at 10 to the minus 10 once. So that comes from that we have such an enormous data set that we will have very good observations occasionally just by chance. So then one can, since we're still working on this, how to exactly do this classification, but one can then see, so for example, if we look at DNA repair proteins, we see that a lot of them have very good evidence. So this is just the beginning of the list. List goes down quite far. So the ones, but they're shown in the order of how strong the evidence is. So these on top here, we always see the ones with the good. And then in the bottom, there are lots of ones that have really very borderline and probably not very strong evidence. And then we have a few cases where we don't observe anything. But you see that out of these 571 proteins, there are very few where there is no evidence at all, but quite a lot where there is sort of borderline evidence. So then this is just looking at the same thing, that we have a p53 binding protein 1, and then we can again,
in this database, when we look at this, all these proteins, if you look at a protein that shouldn't be there, we will always get this very low score.
Yes, I mean that's yeah. So there are the statistics right now, it's a little bit different, but about 80% of the proteins in that so 80% of the genes in ensemble have some evidence that they have been observed. So there are probably about 20% that have never been observed according to this sort of classification. That, and now there are constantly new data sets that people put out, and usually there are very few new proteins being observed. It's only when people look at some very specific cells from some, then there are new proteins that show up. But in general, when people do a random proteomics experiment, there are no new proteins observed. Oh yeah, so this is just going through. So if we now, again, we've looked at this a little bit before, but if you now, so the first view would be that you see the top 20 experiments and what overview of what peptides have been observed in the protein by these different experiments. Then if one digs further down, one can see for one experiment, one can see where, again, the linear representation, but then also representation on the sequence level, and then also showing modifications and all sorts of other information. So these are the descriptions. So you see the sequences and the exon boundaries, and then it shows synonymous and non-synonymous SNPs, Sequences in red are the ones that where it has been observed, and the green ones are the ones that are difficult to observe. Then again, if there are single amino acid polymorphisms observed or are in red, and then chemically modified residues in blue. And as we see here, we see in this case a lot of chemical modification, and that's probably because this is an eye-track sample, so all the lysines are modified, but there are other modifications also. And we see a few single amino acid changes and so on. So then we can go down one level more to see the list of peptides, and then again down also to the spectrum level. So this can be done for all these identifications. They're available on the website. So for 300,000 experiments, for a few million different mzML spectra. So I'm gonna, yeah, now I'll talk about this. So then the other thing that one can see, if one observes a protein, so then one can go in and see how many times different peptides have been observed in the database. So this is the example of catalase. So we have these peptides. So the first one has been observed 2,633 times. And then as these other peptides, they're listed in decreasing number of observations. So what we can also then do is to convert this into a peptide frequency, so that we can see what the frequency is, so that would, if we add up all the peptides that we observed, that should add up to the number of observations. So we add up this column, adds up to 1. And then we can look at how this frequency in the database of observing different peptides is. And if one looks at different proteins, in some cases like this, there are a few dominant peptides, but they're not very dominant, and there's a long tail. Other proteins are going to have just a few peptides, and then it falls down very close to 0. So what we can do then is to, if we have an identification of a protein, then we can go in and get these peptide frequencies from the database and just add them up for the peptides that we observed. And of course, if we observe all the peptides, we're going to get the value of 1. But usually we get something that's smaller than 1, but it should be quite, if we do a good protein identification, we should see something, an omega value that's quite large, because we hopefully are observing these top peptides. So for example, here is just one case where we have observed a few proteins, and then we calculate these omega values for both charge state 2+ and 3+ separately, so that we see that we get high values for most of these. Again, there's a few times, for example, for this one, for 3+, we have a very low value, so probably that's not so probable, but we still have a good value for the 2+. The other thing one can look at is the retention time distribution of the peptides. Here on top, or the red curve is identical in these 3 cases, and that's what we would expect from the whole proteome. If we found all the, if we could see the whole proteome. but usually we see less than that, of course. But here we see quite nice, but we lose some of the the slower peptides of the hydrophobic peptides and a little bit of the hydrophilic, but otherwise we're doing well. But in other cases, we lose much more of the hydrophobic or much more of the hydrophilic. So that's something that the person that does the experiment can use as a tool to make sure that their experiment is well optimized. We can also look at the mass accuracy, and this shows, you probably can't see this, there's actually another curve here. It's probably, you can't see it, but so it just shows that different experiments, you have different mass accuracy distributions, and you want to be as close as possible to zero, and as thin as I mean the width of the curve should be as small as possible. But if you have a well-defined curve up here, of course you can recalibrate everything and move it down. And the nice thing is that if you do that, you can use a much smaller mass tolerance when you search, and you can also use a smaller mass tolerance like Jessica showed you with the MaxQuant. Then other things that you can do is to, for example, project this information, the identified proteins, onto different categorizations, like the gene ontology, cellular processes is what this shows, and it also calculates an expectation value from applying just a geometric distribution onto, and that's a very common way to just calculate how likely this is to be random, that so many proteins from your experiment match a certain category. So this is what then you can do the same thing with the KEGG Pathways. So you again can use a hypergeometric assumption distribution to calculate a p-value for how well again this the proteins that you observe map onto different pathways. And in general, you can do this for any kind of category. If you have, I mean, either the Go categories, KEGG Pathways, but also if you have your own categories, you can apply this kind of analysis to see how much enrichment you get. And then there are lots of software resources, and so Jessica talked about MaxQuant, which is free to use, and it provides a very useful service, and there are lots of other ones also. So, for example, Proteowizard provides a very good resource for converting the starting with converting the raw mass spec data onto more standard formats like the mzML format. And also then doing all sorts of analysis like peak picking and processing of the data. And so this is a resource that a lot of people use. Then there are web-based resources that, for example, Protein Prospector, where you can do all sorts of calculations and searching of databases using peptide mapping data, and the MSMS data, and it's really a rich resource of different tools. There is another resource at Rockefeller that also provides you with many different tools to calculate peptide masses, fragment masses, and so on. So we have developed a tool to easily map if you have found peptides, and then map them back onto the genome to be able to view them in a genomic context and together with other types of genomic data. And also there is the UCSC genome browser, and there are several other genome browsers, but again, so we've used this mapping tool to then be able to take proteomics data and look at it together with RNA-seq and other types of data. Then we talked a little bit about Slice, which is a resource to share mass spec data and share large sets of mass spec data, and without people needing to download the whole data set, but one can go in and extract very specific information. So Slice is for looking at one data set at a time, and then DICE is for looking at searching many larger sets of mass spec data. So then finally, a few words about standardization. There, historically in mass spectrometry, the file formats have been defined by the different vendors of mass spectrometers, and this has caused a lot of problems for especially people that want to analyze their own data and not just use the software that's provided by the vendors. And it took a very long time. I think actually the vendors saw it. They wanted to sell software, so they tried to keep their formats, at least some of them, secret or at least difficult to access, so that they could sell their own software packages. But after a while, it became very clear that the vendors were very good at making really good mass spectrometers, but not so good at keeping up with the software needs. So there was quite a bit of pressure that came from the whole community. And finally, the vendors opened up and said that, okay, we're going to provide libraries so that anyone can read the software. So now it's possible to without buying the vendor software to read and convert it to other formats. And a lot of people use ProteoVisor, but there are other ways to do it also. So, but then the question was, what format to convert it to? So there's been an effort to standardize these, and there has quite a while ago, quite a few years ago, there came out a set of papers about what is the minimum information that's about proteomics experiments that were needed. And this is the main paper that's shown here. And then there were several other. So there is, for example, Miami for mass spectrometry, for mass spectrometry information, for mass spectrometry quantitation, and then for other types of experiments. So then these, for example, so this is for mass spectrometry information. These documents go through and says what information should be given, and some of it is things like when was this data acquired, what software was used, what customization, and so on, and then more description of what the source of the material was, and all these details. So these were the sort of general ideas of what information, but then in parallel there were a few standardized formats that were developed. So these were XML formats, that the idea was that in these, whatever the MyAPI documents described should be represented in these formats. So the ones here that are mostly used, so mzML is used a lot nowadays. But again, it took quite a while before it was adapted. Then mzIdentML is used more and more. So these are the two. The other ones I haven't come across a lot, but they might be adopted later. But right now, the two first ones, the mzML and mzIdentML, is used. So then again, if you look more in detail, the idea is that the mzML should capture mass spectrometry data and preferably the whole complexity that we've looked at earlier, that we have a chromatographic separation, we have MS survey scans, we have fragment spectra, we might have MS to the third, and so all these things should be captured in it. And I think they've done a pretty good job, and it's a good format. And then the mzIdentML then should describe how a protein has been identified. So one should be able to get all the information on how it should refer back, first of all, to an mzML file. And then it should provide all the parameters that are needed for to do the identification, and then it also will contain the identification results that were done. So this is what I wanted to go through today. So the main thing is that there are a lot of different data formats, repositories, and standards. All these are quite a bit in flux still, so it's changing a lot. But for example, on the data repositories front, the nice thing right now is that there are several repositories, and that seem stable, and that hopefully at least have long-term support, so that the community can rely on them, both to deposit their data, but then also to retrieve other people's data and develop new methods based on that.